Methods of food preservation. Good morning, students. Today we will discuss methods of food preservation, which is one of the most important topics in food science and technology. Food preservation plays a vital role in maintaining food quality, extending shelf life, reducing food wastage, and ensuring food safety. In this session, we will understand the principles of food preservation and the major physical, chemical, and biological methods used for preserving foods. Let us begin with the introduction to food preservation. Food preservation can be defined as the science that deals with preventing food spoilage and decay, allowing food to remain safe and suitable for future use. The main objective is to retain food for a longer period without contamination by microorganisms or harmful chemicals. At the same time, preservation aims to maintain the color, texture, flavor, and nutritional value of food. Therefore, food preservation ensures that food remains edible, safe, and nutritious during storage. To understand food preservation better, let us examine its basic principles. The first principle is prevention or delay of microbial decomposition. This can be achieved by keeping microorganisms away from food, which is known as asepsis. It can also be achieved by removing microorganisms or preventing their growth and activity. The second principle is prevention or delay of self-decomposition of food. This involves destroying or inactivating food enzymes and preventing undesirable chemical reactions. The third principle is prevention of damage caused by insects, animals, and mechanical injuries. Together, these principles form the scientific foundation of food preservation. Food preservation methods can be broadly classified into three categories. These are physical methods, chemical methods, and biological methods. Each method uses a different approach to extend shelf life and maintain food quality. Let us study each of these methods in detail. First, let us discuss physical methods of food preservation. One of the most widely used physical methods is preservation by low temperature. The metabolic activities of microorganisms, enzymes, and living tissues depend greatly on temperature. As temperature decreases, the rate of chemical and biological reactions also decreases. Therefore, lowering the temperature slows down spoilage and helps preserve food quality. One low-temperature preservation method is cellar storage. Cellar storage is usually maintained around 15 degrees Celsius. This method is suitable for storing potatoes, onions, apples, and root crops for a limited period. Although spoilage is not completely stopped, the storage life is significantly increased compared to normal room conditions. Another important method is refrigeration or chilling. The storage temperature generally ranges from 0 to 5 degrees Celsius. At this temperature, microbial activity slows down considerably and many spoilage reactions are delayed. Mechanical refrigeration systems are commonly used for this purpose. Products like eggs, meat, poultry, fish, milk, dairy products, fruits, and vegetables can be safely stored for several days under refrigeration. A more effective preservation technique is freezing. The freezing temperature generally ranges from -18 degrees Celsius to -40 degrees Celsius. When food is frozen, water converts to ice. This reduces microbial activity and slows down most chemical reactions. As a result, foods like meat, poultry, fish, ice cream, vegetables, and juice concentrates can be stored for several months. However, some enzymes may still remain active. Therefore, vegetables are often blanched before freezing to inactivate these enzymes. There are different methods of freezing food products. The first method uses cold air or low-temperature gases. Examples include blast freezers, tunnel freezers, fluidized bed freezers, spiral freezers, and belt freezers. The second method is indirect contact freezing. In this method, food comes in contact with refrigerated metal surfaces such as in plate freezers. The third method is direct immersion freezing. Here, food is immersed in or sprayed with refrigerants such as liquid nitrogen. The choice of freezing method depends on the type of food and processing requirements. Let us now move to chemical methods of food preservation. Chemical preservatives are substances added to food to delay spoilage caused by microorganisms. Traditional preservatives include salt, sugar, and smoke. Salt preserves food by drawing water out of microorganisms, causing dehydration. Sugar increases osmotic pressure and reduces water availability for microbial growth. Smoking helps prevent the growth of bacteria and fungi while also improving flavor. Several other chemical preservatives are also used. Acidulants such as benzoic acid, sorbic acid, and lactic acid help reduce microbial growth by lowering pH. Gaseous preservatives such as sulphur dioxide, sulfites, and carbon dioxide inhibit yeasts, molds, and certain bacteria. Antioxidants such as BHA, BHT, propyl gallate, vitamin E, and ascorbic acid help prevent oxidation and rancidity. These compounds significantly increase the stability and shelf life of food products. Food additives also contribute to preservation and quality improvement. Color additives improve product appearance. Examples include tartrazine, sunset yellow, allura red, and brilliant blue. Flavor additives such as nitrates and nitrites help prevent the growth of Clostridium botulinum. Sweeteners such as sucrose and aspartame provide sweetness. Emulsifiers like lecithin help maintain uniformity and stability in food products. The third major category is biological preservation. One of the most important biological methods is fermentation. Fermentation involves the action of microorganisms or enzymes that produce desirable biochemical changes in food. This process not only preserves food but also improves flavor, texture, digestibility, and safety. Many traditional foods owe their unique characteristics to fermentation. A key group involved in fermentation is lactic acid bacteria, commonly known as LAB. These bacteria produce lactic acid, which lowers the pH and inhibit harmful microorganisms. LAB also show antimicrobial activity and help suppress foodborne pathogens. An important example is Lactococcus lactis, which helps prevent spoilage and pathogenic bacterial growth. Thus, LAB contributes significantly to food preservation and food safety. Several LAB strains are commonly used in food fermentation. Examples include Lactobacillus plantarum, Lactobacillus pentosus, and Lactobacillus paracasei. These bacteria can tolerate acidic conditions and survive under challenging processing environments. Their activities improve product quality, enhance preservation, and contribute to the safety of fermented foods. To conclude, food preservation is essential for maintaining food quality, extending shelf life, reducing food wastage, and ensuring consumer safety. The major preservation approaches include physical methods such as refrigeration and freezing, chemical methods such as preservatives and antioxidants, and biological methods such as fermentation. Understanding these methods helps food technologists select the most appropriate preservation technique for different food products. Thank you.